Dear viewers, welcome you all. Today we will discuss about bacterial growth cow. I am Dr. K. Sheila, Assistant Professor, Department of Biochemistry, Sacred Heart College, Autonomous, Tirupattur. Actually, before the detailed classes, just I will show the one diagram. This diagram it's indicate the show that the how to from mother culture, the mother culture they are next to they are subculture. So the mother culture it consists of the sample to be counted. The bacteria they contains high growth of the bacteria will be present. First initially the little for example 1 ml of the bacteria they are diluted to the 9 ml of water, that is a 10 ml, 1 by 10, 10 power of minus 1. After the 1 ml of this solution transfer to another tube, again they are diluted. So first initially they diluted of the dilutions of the tubes, having the initially they contains a high level of the bacteria will be present. So each and every all the subculture of the pipette they contains a less amount and easily count the bacteria growth. So first we'll start the bacterial growth curve. In microbiology, growth is nothing but that refers to the increase in number of the cells. More than 2000 chemical processes were occurred during the growth. The step of the reactions occurs during the growth. Synthesis of macromolecules like a DNA, RNA, polysaccharide, protein, lipid, polymerization. Assembly of macromolecules, cell organelles formation. Growth rate, it increases of bacterial cell number per unit time is referred to as growth rate. Generation, the interval of formation of daughter cells from parental cell is called a generation. Generation time, the time required to form two daughter cells from a single cell is called as a generation time. This is also called as a doubling time. So stages of growth curve. A population growth curve for any particular species of bacterium may be determined by growing a pure culture of the organism in a liquid medium at a constant temperature. Samples of the cultures are collected at a fixed intervals every 30 minutes, and the number of viable organisms in each sample is determined. The data are then plotted on a logarithmic graph paper. The logarithmic of the number of bacteria per millimeter of medium is plotted against time. The bacterial growth curve shows the following four distinct phases. So it is a single bacterium has been inoculated to a liquid medium, and the bacterium starts multiply by binary fission gives the increase of population. Only the single bacterium it need for the helpful for the multiplying of the many number of the bacteria by the use of the process binary fission process. So the four phases are lag phase, log phase or expansional phase, stationary phase, death phase or decline phase. In the beginning, the growth increasing the population will not occur leads to straight line. The lag phase there is no any increases of the population. Then the cell starts multiply gives linear line like a log phase or exponential phase, followed by straight line that is a stationary phase, and the finally decline line linear line like a death phase. So first initially there is no any increases it will occur in the first initial phase, it's a lag phase. It's also straight line. Then the cell it start multiply with the help of the nutrient, so it gives a linear line. They may be the cells or little multiply. that is called as a log phase or expansional phase, followed by straight line like a stationary phase and decline linear line or death phase. So first one we will discuss lag phase. When a bacterium is introduced into a medium, it will not multiply immediately. After a period of time, only the bacterium will start multiplying. This period is called as a lag phase. During this period, the cell increases its size. It will synthesize new protoplasm and enzymes. The bacterium will adjust the physiological environment. At the end of the lag phase, cell will start reproduction. Log phase or expansional phase. After lag phase, the cells start multiply steadily by binary fission at constant rate. The portion of curve or linear and have constant generation or doubling time. All the cells are uniform chemical composition, metabolic activity and physiological characters. At this time, the generation time will be of constant. This is the ideal stage to study the metabolic activity of the bacterium. Third one, the stationary phase. By this time, the essential nutrients of the medium get depleted and some toxic substance produced by the organism leads to slow down the cell division. So first phase, the lag phase, there is no any increases the bacteria. The second phase, the log phase, the straight line of the linear line will be formed. The cells will start the multiply. But the stationary phase, the essential nutrients of the medium get depleted. Some toxic substance will be produced. So this toxic substance, how do they are produced? Means the by the organism leads to slow down the cell division. At this time, no net increases or decreases of cells in the medium. It occurs. So this stage is called stationary phase. So this stationary phase, there is no increases or decreases of the cells. During this stage, cell division rate and cell death rate is uniform because of there is no net charges of increases as well as decreases in the cells. Cell death occurs due to low nutrients content, pH change, toxic waste produced and oxygen content reduced. So final one, death phase or decline phase. In this phase, few cells are alive and few cells are died. The death rate is higher than the growth rate, leads to decline the population. So this phase is referred as decline phase or death phase. The cell lysis occurs during this stage. Some species can die very rapidly because there is no, some only few of them they are alive. So their species of die it's very rapidly and some can persist for months. So these are the bacterial growth curve. Thank you all.